Hi guys, it's Dr. Buhler, and we're going to talk a little bit about neurotransmitters, my favorite things. We've got several abbreviations. Normally when we talk about neurotransmitters, we use the full name, but when we write them, we use the abbreviation. So you're going to see me writing out these abbreviations quite a bit. You guys already know that there are two receptor subtypes that are normally used for neurotransmission. Now, neurotransmitters don't generally use hormonal receptors, tyrosine kinase receptors, none of that. The only ones we're going to be looking at are the ligand-gated ion channels and the G protein coupled receptors. There are a few different terms we use for these, but ligand-gated ion channel, sometimes people call them ionotropic because of ions. And for G protein coupled receptor, that's normally what we're going to call them, but you can also hear them referred to as metabotropic. Yes, that's my cat, Oreo. So what we're going to talk about now are going to be some of the major neurotransmitters, and there are even more neuromodulators, which you can think of as acting more like allosteric compounds. But we've got several structural families of neurotransmitters. We're going to talk about those. We're going to talk about the receptor types that they have, some of the really basic effects on cellular activity, mostly focusing on whether they're excitatory to neurons or inhibitory to neurons. And then we're going to talk about some of the basic examples, I would say, of physiological effects and associated disease states. So with every neurotransmitter, there are so many different systems it's involved in, so many different physiological effects that we can't say that we're going to even close to address all of them here. And even after the whole 2 years of your curriculum, you still won't have addressed all of them. But I want to give you some kind of conceptual framework because I'm going to be throwing a lot of neurotransmitter names at you, and it's just going to be words if you don't have some type of framework of types of disease states that they're associated with. One of the other things we're going to talk about, and I think this will make a lot of sense to you about what we talked about with enzymes and with agonists, that the receptor itself matters and the enzyme itself matters, but where it is matters a lot as well. So we have this certain finite number of neurotransmitters. We have even more neurotransmitter receptors because most neurotransmitters have multiple subtypes of receptors. And then those can go on different neurons and different combinations in different brain regions. Some of them can be on the outer dendrites, the inner dendrites, the cell body of the neuron. We even have receptors on the axonal terminal of the neuron. So all these different combinations of receptors and location, and even what the cell does internally in response to that signal, is going to be very different in different systems. So what I'm really getting at there is we may talk for an hour about how dopamine acts on motor systems in Parkinson's and all the different ways we can modulate that. But then we might go to a different disease state in a different brain region, and we're still talking about a dopamine neuron, but it's modulated very differently. So that's really what I want, is to make sure that you recognize that while we're talking about these neurotransmitters almost as if they're always going to have the same effect everywhere they go, that's not in reality the case. And then we're just going to review very quickly some of the mechanisms for receptor tolerance. We have some learning objectives. Overall, we're going to look at those two types of receptors, those ligand-gated ion channels and the G protein coupled receptors. For those neurotransmitters, know the families, those example functions, what those receptor types are. And that's going to be a big focus of what we're going to talk about in terms of some of the major subtypes for some of the major neurotransmitters, and then some of the cellular activity. We're also going to have a moment where we're really going to talk specifically about a class of neurotransmitters called the monoamines. And that's because we have three really common neurotransmitters, norepinephrine, serotonin, and dopamine, as well as a derivation of norepinephrine, epinephrine, also known as adrenaline, which are all structurally in the same group. Because of that, when we talked about receptor specificity, we said that we have that certain shape, conformation of the receptor binding site. If there are multiple different ligands that have very similar structure, they're all going to have some affinity for that binding site. So when you start looking at medchem and you start looking at drugs that act on noradrenergic or norepinephrine, serotonergic, and dopaminergic systems, it's really important that we recognize that there's going to be some cross activity at receptors and at degradatory enzymes for all three of these. Then we're going to mention a little bit about how peptide neurotransmitters work. And we already talked about learning objective number 5, which is how the effects really depend on the system you're in. So we already mentioned that really the two types of receptors we're looking at are ligand-gated ion channels and G-protein coupled receptors. And again, sometimes people call those metabotropic. I am not going to generally call them that, but I need to tell you that because for at least one neurotransmitter, that's the subset of receptors are called metabotropic. So before I even start talking about these specific types of receptors, I want to make sure to really hit home something that is always very problematic and confusing for students. These guys, ligand-gated ion channels, are ion channels. And so I'm going to draw them over here, even though this is closer to the G proteins. So these are ion channels, but most importantly, they are receptors, which means they bind a ligand and they open up. The ion channel part of it is just, that's the effect when it opens up. We have other ion channels as well. You saw those in action potentials. Those were voltage gated. Those were also ion channels, but they're completely unrelated to the receptors. Voltage-gated ion channels just happen to also gate ions. So gate is also a term we use when we mean let through. So voltage opens the gate for ions to go through on those particular ion channels. And if it's a receptor, it's ligand-gated, which means the ligand is what opens the gate for the ions to go through. So that's one really big thing, is know the difference between voltage-gated and ligand-gated. The second thing to really make sure that you understand about these ligand-gated ion channels they are a receptor that binds the ligand on the outside of the cell. It never lets the ligand in. The ligand is not passing through that ion channel pore. So the ligand is going to stay outside. We can see that over here. If I make a ligand here, binds to that ion channel receptor. Now, in this case, sodium can go through because that binding caused that conformational change, opened up, sodium went in. But that ligand itself, so those were the two really important things to remember. That voltage-gated ion channels and ligand-gated ion channels are not the same thing, and that with a ligand-gated ion channel, the ligand does not enter the cell. So a little bit more detail on these different types of receptors. You're going to need to know that for ligand-gated ion channels, we really have a small group of ions that are going to come in and out. We have ones that let sodium in, ones that let calcium in, and those are both going to excite a neuron. So when we say excite, we mean it's depolarizing. It's going to make it more likely to fire an action potential. And then you have channels that let chloride in, and that's going to inhibit the cell, or that let potassium out, and that's also going to inhibit the cell. And again, inhibit because it's hyperpolarizing, whereas this was depolarizing. So there are multiple different types of G proteins, and each of them are going to have very different cellular effects inside. We've got GS, GI, GQs, GOs, G11s, multiple different ones. If you're a cell biologist, you're going to learn all of the effects that happen from activation of all of these different G proteins. What we're doing for neuro is really we want to know, is this activation going to excite a neuron and cause it to be more likely to fire, or inhibit a neuron and cause it less likely to fire? So the two that we are most focusing on are the GS, and S is for stimulatory. That's going to increase cyclic AMP. Those are going to excite a neuron and make it more likely to fire. And GI, I for inhibitory, those are going to decrease cyclic AMP, and they are going to make it less likely for a neuron to fire. While the ligand-gated ion channels had pretty close to only one effect, which was ion voltage change, the G protein receptors have a multitude of intracellular effects. So you know that those are going to change cyclic AMP, PKC, inositol triphosphate, DAG pathways. There's phosphorylation of everything. Some of them are going in and grabbing calcium out of the intracellular calcium reserves. So when we look at G protein coupled receptors, there's a lot more going on downstream than there is for ligand gated ion channels. And the other thing that I want to tell you here is to make a point that the endogenous ligands that we have in our body are agonists. They're not antagonists and they're not inverse agonists. Some of them can be partial agonists, some of them are full agonists. But the reason that they're always agonists is because when our neurons are here in a synapse, here's the neurotransmitter, here's the receptor, we're always starting with no ligand bound on that receptor. So that receptor here is empty and it's off. The body isn't going to have a neurotransmitter that's an antagonist because that's already off and there's nothing else that would be an agonist that could bind in the normal body system, the way we evolved without any drugs coming on board. So if this is already off, the only thing you can do is activate it in order to get a response. So all of the endogenous agents that we have, our neurotransmitters, most of our hormones, there's always going to be exceptions to each of these statements. But basically, you're considering these receptors off until the ligand hits. So they're all going to be agonists. So when we're looking at drugs, we can throw in a drug agonist to activate a synapse that hadn't been activated before, but that we want to bring into the picture, or we can give an antagonist to block the endogenous activity, to block what your body is naturally doing with releasing the neurotransmitter. Okay, this is a big slide. This is a very densely written, but actually pretty concise overview of what we're going to be talking about in the next slides. So again, these are the major neurotransmitters, pretty much all the neurotransmitters. Some people call out the agents, the neurotransmitters that are peptides, and refer to them specifically as neuropeptides. I won't be doing that from here on out. If it's acting as a neurotransmitter, I'm going to call it a neurotransmitter, and we're only going to mention that it's a neuropeptide if that comes up specifically needing to know that. So we're going to have a slide for each of these neurotransmitters, but I'm just going to orient you here. We have acetylcholine. One of the main functions that you know about acetylcholine is autonomic nervous system. Also acts in the central nervous system, but when we think about acetylcholine after we take first semester A&P, that's kind of a key for your brain to kind of hit on and go, oh yeah, acetylcholine, that helps mediate the autonomic nervous system. We'll also see that it's involved a lot in learning and memory. We have glutamate. What I want you to know about glutamate is it is the primary excitatory neurotransmitter. And I mean, you know that. I want you to be able to just wake up in the morning and go, glutamate is the primary excitatory neurotransmitter, and GABA is the primary inhibitory neurotransmitter. So if it's excitatory, it means it's going to depolarize, and it's going to help fire action potentials. And if it's inhibitory, that means it's going to hyperpolarize and inhibit action potentials. And when we look at what glutamate and GABA do physiologically, they kind of make sense if you're thinking about, are you activating action potential generation and neuronal excitability on a broad scale? So if you're activating a lot of activity, maybe that could increase seizure events. They could increase migraine events because those are based on having a focus of overactive neurons. On the other hand, if you're reducing general excitation, maybe you could help use agents to reduce seizure. These agents are going to reduce this overactivity. Maybe they can reduce anxiety. They could cause sedation. So when you learn what these neurotransmitters do cellularly, then some of the reasons, physiological effects and reasons that we use drugs start to fall into place. Our next one are norepinephrine and epinephrine. And what I want you to know is, here, let me draw it up here. Norepinephrine is the same thing as noradrenaline, and epinephrine is the same thing as adrenaline. And that is because no one can agree on anything. So actually, originally, there was a difference between British and American terminology. And then what ended up happening was all that got mixed up with each other. And normally, and you're talking about synapses, and using these as a neurotransmitter, we say norepinephrine. If we're talking about it being used as a hormone, we may call it epi, or we may call it adrenaline. At the synapses, while we call the neurotransmitter norepinephrine, we often call the system adrenergic for noradrenergic. So it's all a big mess. So if I'm talking about an adrenergic system or a noradrenergic system, we're talking about adrenaline or norepinephrine. So if I call something an adrenergic system or a noradrenergic system, you know that we're talking about adrenaline being the same thing as epinephrine and noradrenaline being the same thing as norepinephrine. Norepinephrine is that second main transmitter in the autonomic nervous system. Think adrenaline, so fight or flight. But it's also going to be involved in executive function and cognition and keeping things going and moving right up there. And just like acetylcholine, even though we're kind of pointing out the ANS, it's also involved in the CNS. Dopamine, we often think about dopamine in terms of reward and addiction because all addictive agents and activities cause huge dopamine surges. If you do something, whether it's take heroin, gambling, some people running does this for, have sex, eat chocolate, all of these are pleasurable activities, but they're not just like a calm pleasure, like, oh, that was nice. They are something that gives us reward. And then that tells our body, oh, hey, you got a huge dopamine pump. Here's reward. Now you really want to do that again. So it's our body's way of making sure that we focus on things and focus on doing things that make us feel good. Dopamine is also really involved in depression. And so we're going to see that we're going to have some antidepressant agents are also going to have dopamine activity. Looking at this, you can see that dopamine has really widespread effects in a lot of different disease physiologies. So this isn't saying that dopamine is more important or even more widespread in physiology than the others. It's just it seems to be involved in a lot of pathophys and a lot where we have drug targets. So we talked about depression, schizophrenia, motor control, pleasure and reward, which can turn into addiction, helps with cognition and ADHD. And one thing that is important, and we'll talk about in the autonomic nervous system, is that we do also have our own nervous plexus in the GI tract. And some people call that a third arm of the autonomic nervous system. Like you have sympathetic, parasympathetic, and GI, or enteric nervous system. And there are a lot of neurotransmitters there onto the parietal cells, onto smooth muscle, etc., that are going to have effects on your GI function. And dopamine also can cause nausea and vomiting. So those are the main things that you need to know right now just to get that conceptual framework. 5-HT or serotonin. When we think about serotonin, one of the first things we think about is what are the major antidepressants? And we think about SSRIs. So selective serotonin reuptake inhibitors, if you inhibit reuptake, then you don't take it back up. That means it stays in the synapse. So that means you increase, those are arrows there. You increase your serotonin levels. And that helps treat depression. So serotonin is very involved in mood and pleasure. Also involved in migraine, have drugs that act at serotonergic receptors for migraine treatment, also causes nausea and vomiting. Opioids are actually neuropeptide. You probably actually know a lot about how opioids act just in terms of common knowledge. They relieve pain. They can also cause sedation and at higher doses respiratory depression, which can be lethal. Cannabinoids are a lipid structure. They're going to relieve pain, but they also have receptors in the learning and memory areas of the brain, the hippocampus. You don't have to memorize hippocampus. Not yet. And histamine, which acts both as a neurotransmitter in the nervous system, so you can have neurons that are releasing histamine, but it can also be released by cells outside of the nervous system. So I'm going to try to draw a little mast cell in the allergic immune response. And if it releases histamine, then that's acting in more of a paracrine or autocrine function. So some of these, specifically adrenaline and histamine, can act more hormonally or autocrine or paracrine, as well as the systems where they're neurotransmitters. So histamine actually promotes that allergy response. You release histamine out in your tissues, you get itchy, red, swollen. That's why we use antihistamines as anti-allergy. And also increases nausea. So a little bit more detail about these neurotransmitters. Here, what we're really going to focus on, we have the same example functions here, but I'm going to focus on what the family is and what the receptor subtypes and types are. So acetylcholine is one of the most basic neurotransmitters. It's on its own. We don't have another neurotransmitter in that family, so it's just acetylcholine. I have the structure here just because it's really cool to see structures, but you don't have to memorize the structures for any of the neurotransmitters for me. What you do need to memorize is you need to know the receptor types that acetylcholine acts at. So acetylcholine has both ligand-gated ion channel and G-protein coupled receptors. When we talk about the ligand-gated ion channels, they are nicotinic ion channels. So if I say you have a nicotinic receptor, then you know that that receptor is activated by acetylcholine, and that we're talking about a ligand-gated ion channel, and that's nicotinic. If I say you have a muscarinic receptor, then you know that that is activated by acetylcholine. It's a muscarinic receptor, which is a G-protein coupled receptor. One of the things about the ligand-gated ion channels here, the nicotinic receptors, is that they're always excitatory. There are a few different types of nicotinic receptors. I'm going to circle nicotinic here. There are a few subtypes, but what you're going to notice is once acetylcholine's bound and they open up, they're all going to gate in. Remember, gate is open up and let in. Calcium and or sodium. Some of them let in mostly just sodium, some of them let in a lot of calcium as well. So that sodium and the calcium are both positively charged. So you're thinking, okay, this is depolarization. And you are correct. If we're just talking about sodium coming in, that's pretty much all it is depolarization. But when you bring in calcium, you actually can have more complex and modulated effects as well. The first thing that's going to happen is, yes, you get your depolarization, but then if you think about your Dr. Roberts and your prereqs and those signaling transduction pathways, a lot of things got altered when you had calcium levels changed. So with one of these nicotinic receptors, you might bring in calcium and sodium, have immediate depolarization, firing action potential, and then that calcium could act downstream at additional systems and cause changes in how that neuron responds. And we're going to see that when we talk about Alzheimer's and we talk about pain, that these receptors bringing in calcium can make long-term changes to neurons that are detrimental to us. So we have those nicotinic receptors coming back up here. Those are sodium, sometimes also calcium, always excitatory or depolarizing, always ligand-gated ion channels. Fun fact, the reason we call them nicotinic is because when we first discovered them, we discovered them by pouring nicotine onto the tissue, and that's what activated that subset of receptors. When we talk about ANS, we're going to talk about a lot of nicotinic receptor types. Well, we're really only going to talk about 3. We're going to talk about the neuromuscular junction nicotinic receptors, which are often referred to as NM. We're going to talk about the ganglionic nicotinic receptors, which we often refer to as NG. And we're going to talk about the CNS nicotinic receptors, which are usually NN for neuronal nicotinic. So those will come up very soon in the next lecture. So while I said that for acetylcholine, nicotinic receptors are always excitatory, it's not going to be that simple for muscarinic receptors. We have multiple different subtypes of muscarinic receptors. Here we have M1, M3, and M5. These guys have a GQ coupled pathway. We have M2 and M4, and these guys have a GI coupled pathway. GQ, remember I said that we're going to just stick with GI as inhibitory and GS as stimulatory because these other subtypes just aren't so clear. So when we talk about those, I'm just going to say varied effects. And then here I'm going to say inhibitory effect. And then I'm going to say stimulatory effect for these. All right, I think we got acetylcholine. Here I'm not going to talk about the breakdown. So this is not breakdown information here. Breakdown is super cool. I love it for all the neurotransmitters, but we'll be talking about that when we go into different disease states because a lot of our drugs, while a lot of our drugs are agonists at a receptor, antagonists at a receptor, a lot of our drugs are also inhibitors of breakdown enzymes. So those will be coming up later. So glutamate. Glutamate, as I said, is the major excitatory neurotransmitter. It's structurally really simple. It's actually just an amino acid. So we said this was the primary excitatory neurotransmitter, and these were some examples of what that excitation of the system can do. Here we also have two receptor types. We have ligand-gated ion channels again and G-protein coupled receptors again. Our ligand-gated ion channels here have some very specific subtype names that I'm not going to have you learn right now. So all I would be referring to these right now would be to say glutamate ligand-gated ion channels, if that's what I'm talking about. When glutamate binds to these ligand-gated ion channels, it's going to conformationally change, open up, and what we're going to see is mostly sodium coming in. In the case of this subtype, we also see calcium. We're going to talk about that later when we talk about Alzheimer's. So when we're thinking about these right now, we're thinking about sodium coming in. That's depolarizing. So that's going to be excitatory and fire action potentials. When we're talking about the metabotropic glutamate receptor, that's the G-protein coupled receptor, M-GLU. Remember, metabotropic was just another way of saying G-protein coupled. These receptors can be coupled intracellular to different G proteins. So again, these are kind of more complicated. You have varied effects. So you've got GOs, you've got GQs. You're not having to memorize that. So we're going to say here that the effects are varied on the cell. GABA, which is one of my favorite neurotransmitters, is also an amino acid. It is the main inhibitory neurotransmitter, has a lot of tone throughout the body, throughout the CNS. And what I mean by tone is almost every neuron has some amount of GABAergic input onto it. So by giving a GABA agonist, you're going to really be reducing the activity of a lot of systems. And I mentioned some of the systems that we look at therapeutically. We're looking at things that may have pathophysiologically have overexcitation, and then glutamate be able to bring that excitation down to give us a beneficial effect. You're not going to be surprised. We have ligand-gated ion channels and G-protein coupled receptors. The terminology here, very simple. If it's a ligand-gated ion channel, it's called a GABA-A receptor. And if it's a G-protein coupled receptor, it's called a GABA-B receptor. So A, because ligand-gated ion channels are better, and B, because G-protein coupled receptors are just, kind of not as cool. So you don't need to know anything more detailed about the structures for any of these receptors in any more detail than I'm actually talking about. So as I talk about this GABA-A receptor, you're going to know that GABA binds, and you're going to want to know that chloride comes in and that becomes hyperpolarizing to a neuron. But you're not having to memorize right now all these other allosteric sites or anything like that. With GABA-B, you need to know that it's a G-protein coupled receptor, GI coupled, because that's the inhibitory G-protein. Again, you don't need to memorize this structure, but I just want to point out that if this looks like a really weird G-protein coupled receptor, it's because it's actually a dimer. You have two GABA-B receptors that have come together into a functional dimer receptor. So to review that, GABA, an amino acid neurotransmitter, primary inhibitory. If you forget and are thinking, oh, what does that channel gate? I can't remember. Is it sodium? Is it chloride? Well, what does this do? It's inhibitory. And so if it's inhibitory, you mean you want to hyperpolarize. Chloride coming in is a way to do that. That's going to hyperpolarize. So you had those two subfamilies, ligand-gated ion channel, GABA A, G protein coupled receptor, GABA B. And that's going to be GI coupled. So monoamines. Monoamines are really cool. And this is when it really does matter to understand the structures and understand how these are related to each other. So when we talk about monoamines, we're talking about serotonin, 5-HT, talking about dopamine, and we're talking about norepinephrine. So all of these, you're not going to be surprised, one amine group. Structurally, these are all really similar. And so we're going to have some of that crosstalk or that non-specific and non-selective action at receptors and enzymes from these three. So you've got to know your nomenclature here because we're going to talk about drugs and you're going to need to know what's a monoamine, what's a catecholamine. So all of these are monoamines. So your serotonin, your norepinephrine, and your dopamine are all monoamines. However, if you kind of notice here, serotonin definitely looks different than dopamine and norepinephrine. So these are also catecholamines. And they actually look so similar because dopamine is actually the precursor to norepinephrine. So if you're in a neuron and that neuron wants to be a dopaminergic neuron and release dopamine as its neurotransmitter, then it's going to synthesize dopamine and it will not have the enzyme needed to make norepinephrine. So it's all going to stay dopamine. However, if you're in a neuron that wants to make norepinephrine, you're going to synthesize dopamine, but that cell is actually going to have that enzyme. Therefore, it's going to drive all the dopamine into norepinephrine, and that's what you're going to end up with. So even though norepinephrine's made from dopamine, any individual neuron is only going to release one of them. We're not having neurons that release both. So pharmacologically, one thing that really matters about knowing that serotonin, norepinephrine, and dopamine are all monoamines is that they're all broken down by the same enzyme family. That's monoamine oxidases. And how do you remember when I ask you in a couple weeks, what neurotransmitters do monoamine oxidases break down? Well, you're going to think it breaks down monoamines, and I know that my monoamines are norepinephrine, dopamine, and serotonin. However, then I'm going to ask you in the second part of the question, hey, what neurotransmitters are broken down by COMT? And COMT stands for catechol O-methyl transferase. And you're going to say, oh, okay, that's the one that breaks down catecholamines. So that's only going to break down dopamine and norepinephrine. So you're going to see drugs that are specific to inhibiting COMT, and you're going to see drugs that are specific for inhibiting MAOs. And you're going to be able to know what different neurotransmitters are going to be affected by those different enzyme inhibitors. So just distinguishing between monoamine oxidase and COMT is obviously not complicated enough. So now we're going to say, oh, within monoamines, we're going to have different subtypes of those. When we talked in the first lecture that we did on enzyme action, I used monoamines as an example of enzyme activity. And so those were the enzymes that broke down these monoamines. And I mentioned that there were two types that we talked about, monoamine oxidase A and monoamine oxidase B. And I said at that time you didn't need to memorize those enzymes. Now you do. Because monoamine oxidase A and B are so similar to each other, all three of these neurotransmitters can bind and be broken down, but not at the same affinity. So monoamine oxidase is very active at breaking down noradrenaline and serotonin, and it breaks down just a little bit of dopamine. But monoamine oxidase B is very good at breaking down dopamine, and we're kind of ignore tyramine here. So this selectivity matters in terms of drug development, because if you want to increase noradrenaline, serotonin, and dopamine, then you're going to want to make an inhibitor that inhibits both A and B. Whereas if you only want to inhibit dopamine, then you're going to want to make a drug that pretty much is selective toward inhibiting MAOB. So that's something that I want you to really get a handle on, is this neurotransmitter family of monoamines and how they break down and why that's important. And I think we already talked about this. We talked about the monoamines themselves. We talked about the catecholamine subset, dopamine, norepinephrine, and epinephrine. Epinephrine being the same thing as adrenaline. I'm not going to go through the functions here. I say, because we already talked about those, when I say modulates, what I'm trying to say is it's not specifically stimulatory or inhibitory. So really that means it has varied effects in cells. And the same thing for norepinephrine and epinephrine. These receptors are modulatory, so they have varied effect intracellularly, depending on what G protein is being activated. And here, for the catecholamines, these only have G protein coupled receptors. So these do not have ligand-gated ion channels. There are several different subtypes of dopamine receptor, and I just write R for receptor because you're going to be writing the word receptor a lot, and you don't want to have to spell it out every time. So there's several different subtypes, but we're not going to worry about those till we get to those disease states. However, it's much more important to know up front about the adrenergic receptors. So a little terminology here. When we have a neuron, we're releasing the neurotransmitter onto another neuron. Whatever this neurotransmitter is that's being released, this cell is going to be ergic for that neurotransmitter. So to say that a little bit more clearly, say we have dopamine being released here. That means this cell body is the cell that made that dopamine. So this is a dopaminergic neuron. So dopaminergic neuron, dopaminergic synapse. And even though you know this postsynaptic cell's got to have dopamine receptors on it, that doesn't make it a dopaminergic neuron. You don't know what that one might release. That might release GABA, it might release serotonin. That would make it either a GABAergic neuron, because that's what it would release, or a serotonergic neuron, if that's what it releases. But you're always naming the ergic based on the neurotransmitter that that cell makes and releases. So that being said, remembering that norepinephrine and epinephrine also have that terminology of adrenergic or adrenaline. When we talk about these receptors, we're calling the whole batch adrenergic receptors. Those alphas and beta subtypes are really important, and you're going to have to just have those memorized as well. The adrenergic subtypes, the alpha subtypes, bind both norepinephrine and epi, but they bind norepinephrine at a much greater extent. Beta is going to bind epinephrine at a much greater extent. Alpha 1 is a GOGQ, so it's going to have really varied effects in a neuron or in an effector tissue, whereas alpha 2 is GI coupled, so it's going to have those inhibitory effects. For the beta receptors, all of these are GS coupled, so they're all going to be excitatory. Yay! Something just made your life easier. So let's step back and see what that matters for. You're releasing norepinephrine or epinephrine. Norepinephrine is more likely to be involved in synaptic transmission, and epinephrine, known as adrenaline, is more likely to be involved in kind of a more hormonal type function. Well, actually not more hormonal type, but as a hormone. You're releasing adrenaline through the adrenal cortex, going through your whole bloodstream, and that is going to then bind to and act on any adrenergic receptors that it hits in your entire organ systems. So norepinephrine is more likely to be synaptic, epinephrine or adrenaline more likely to be hormonal. So these alpha receptors are going to be preferential for norepinephrine. The beta receptors are preferential for epinephrine, so it won't be really surprising to you to see that these beta receptors are going to be on a lot of organs that are activated hormonally by adrenaline release. So I'm going to say systemic activation. Okay, we're good. Serotonin, I think we already said most everything there is to say about this. It's a monoamine. It is not a catecholamine. So it is broken down by monoamine oxidase and is not broken down by COMT. Again, quite varied effects, modulates neurons. There are both ligand-gated ion channels and G protein coupled receptors. At this point, I am not going to have you guys learn those subtypes. There is only one ligand-gated ion channel serotonin receptor that we have a drug for, and we'll talk about that when we talk about migraines. When we take drugs to increase serotonin signaling, we're rarely taking an agonist. We're usually doing things like blocking enzyme breakdown so that we bring up serotonin levels through natural synapse release. So because of that, we aren't going to be designing drugs that are specifically acting at only one subtype of G protein coupled receptor. So basically, that was a lot of information to say that this isn't interesting enough for us to really break down. Please don't tell anyone who specializes in serotonin that I said that. So some of the other neurotransmitters I'm going to write here because this might be a little confusing. Neurotransmitters, those can include our amino acid neurotransmitters, acetylcholine, monoamines, and we have neuromodulators that were more those allosteric. In this lecture here, we're not going to discuss these neuromodulators. They are going to come up in later lectures. Don't write this down, but there are things like substance P, CGRP, nitric oxide, various different things that are going to act allosterically. But the three neurotransmitters that you do need to know about right now that kind of stand off from the ones we've already talked about are opioids, which are a neuropeptide. And again, I think I said that the neuropeptide neurotransmitters, they're coded for in the genes, they get transcribed, they get translated into a longer peptide, proteases break it up, you get into a neuropeptide, that gets transported down to the presynaptic terminal, and there you go, you got your neuropeptide neurotransmitter. Opioids only have G protein coupled receptors, and all of these receptors are GIO coupled. So I've been kind of making it simpler and just saying GI. I could also say GI-GO. Same thing. So if it's all GI, then it's all inhibitory. Same thing with cannabinoids. Cannabinoids are all GIO. And so they're all going to be inhibitory. So any cell that has an opioid receptor or cannabinoid receptor is going to be inhibited if that receptor is activated. We also have another amine, but it's not as related to the monoamines. So we generally kind of pull that out also because it acts really differently, as in really different systems. So we generally pull that out separately and talk about histamine. It also has only G protein coupled receptors. It's a lot more complex. We have varied G proteins, so really varied responses. It has both neurotransmitter and autocrine or paracrine type effects. And we already talked about that. So one of the key concepts then, after knowing about these major neurotransmitters, was that idea that we have this limited number of neurotransmitters, but we have a lot of subtypes, and we have a lot of different pathways in our body, and we have a lot of different G proteins and downstream kinases and modulators of those G protein receptors. So with mixing all these things together, there are no two synapses that are the same. So if we're talking about a pathway for dopaminergic control of motor systems, and I say, oh, here's receptors that modulate this presynaptically and change the dopamine release, and these are the things that happen postsynaptically with these D2 receptors, and then I move and I talk about a whole completely different system that has dopamine receptors like the GI system, you're not going to have the same things modulating it. So that's just a really important thing because sometimes we get so detailed about a specific pathway when we're talking about a drug target, and we talk about all these different ways we can modulate that, and then we move to another system and a lot of times people think, oh, we'll modulate that the same way as we modulated the first one, but that's not always the case. So that's there to make things more difficult. And then super briefly, I know you talked about tolerance already, and tolerance was just when you've been taking an agent and your body responds to that in a way to try to create balance again, get closer back to the normal activity. So when we're looking at neurotransmitter receptors and drugs that act at those, and we look at tolerance, we can look at what happens when we put that drug in your system outside of your brain. So are you changing your CYP450 metabolism of it? If you've changed your blood pressure with the drug, do you have other pathways that affect blood pressure that are just going to come in and take over instead? So there are these larger physiological methods to overcome a drug effect and create tolerance. But we're really going to see, when we're talking about effects on cells, we're talking about the changes to a receptor. And there are a few different ways you can desensitize the receptor itself so that once ligand binds, it's less likely to activate, less likely to turn on. That can happen really quickly. You phosphorylate the receptor, now it doesn't work anymore. A second later, you want it to work again, you dephosphorylate. It's that simple. If you want something to last a little longer, you can endocytose a receptor, hang on to it for a while, and then put it back onto the membrane when you need it. Or if you really want a long-term effect, you can just completely downregulate that receptor transcription and translation. So I think that is it for our basic overview of the major neurotransmitters. And I'm really looking forward to talking to you guys about some of my favorite neurotransmitters and my favorite pathophysiological pathways.